Hello, I am Selvi Thiruvengadam. I am the narrator of the following presentation on behalf of my father, Alagu Thiruvengadam. This presentation summarizes my research over 20 years, starting from 1998 when my son was diagnosed with bipolar disorder. It covers diagnosis, treatment, scientific aspects, and new drugs for bipolar disorder and ADHD. I just retired at that time after teaching and practicing mechanical engineering. When I got started, I considered living cells as an electromechanical biological system, as shown in this figure taken from the cover of Science magazine. The cell components are contained in a semi-permeable membrane. If you measure the voltage across the membrane, you will observe a voltage. This is called the membrane potential, or MP. This potential arises from the ionic gradients across the membrane. For example, sodium concentration is about 18 millimole inside the cell and about 150 millimole outside the cell. Similarly, potassium concentration is about 135 millimole inside and only about 3 millimole outside the cell. Calcium and chlorine have similar gradients, but their contribution to MP is small because of their low permeabilities. Potassium has 2 orders magnitude higher permeability than that of sodium. The primary contribution to MP comes from the potassium gradient because of its higher permeability. Lithium permeability is close to that of potassium. I am familiar with the electrochemical properties of sodium, potassium, and lithium alloys since I have worked on the space nuclear auxiliary power systems during the early years of the U.S. space program. I became interested in the lithium efficacy in bipolar disorder as I began my research. What happens to MP if we add lithium to this milieu of ions? I used the neuroscience equation called Goldman-Hodgkin-Katz equation to deal with multiple ionic gradients. These calculations showed that lithium would depolarize the membrane potential. When I sent it out for publication, one of the reviewers referred me to the clinical observations of L. Malik and others, which supported my results. The top right figure shows my calculations, and the lower figure shows the clinical observations. These observations showed that the MP was hyperpolarized in premedicated bipolar patients in the hospital, and the MP was depolarized close to those in controls and in euthymic patients on lithium. These clinical observations lent credence to my calculations. I showed these results to my friend Dr. Krish Chandrasekharan. He invited me to his molecular biology lab at the University of Maryland School of Medicine. We measured the MP in cultured lymphoblasts collected from bipolar disorder patients, their siblings, and matched controls. These cells were obtained from the Coriell repository operated by NIH. The results are shown in the box plots on the right. The bottom border of the box shows the 5th percentile, whereas the top border shows the 95th percentile. The MPs in bipolar patients were significantly different from those in siblings and controls. This result led us to consider MP as a diagnostic marker. We developed an assay to measure the MPs in whole blood cells from patients and controls. The measurement of MPs is a complex and tedious procedure. We developed a ratiometric assay using 2 buffers to simplify this procedure and make it readily usable by lab personnel. The assay consists of 2 potassium-free buffers. One is called the reference buffer, containing sodium, calcium, and glucose at physiological concentrations, while the other is called the test buffer that contains ethanol added to the reference buffer. The ratio between the MP in the test buffer and the MP in the reference buffer is called the membrane potential ratio, or MPR. We conducted a clinical trial using this assay at the psychiatric department of the University of Maryland's Medical School. We included patients with schizophrenia and depression, in addition to bipolar disorder and controls. As shown in this slide, the NPR values in bipolar patients were significantly lower as compared to other groups. All the patients were hospitalized adults. We decided to expand this trial to a larger population representative of the community as a whole. I set up a lab at Dr. Murphy's clinic in Columbia, Maryland. Dr. Woodruff joined us at this effort. We included ADHD patients in this trial. We found that the NPR in the BD group was significantly lower than that in the negative group, while the NPR values among the ADHD group were significantly higher than that in the negative group. Negatives include controls and other psychiatric patients with no known BD or ADHD symptoms. We completed about 1,000 samples. Dr. Woodruff contributed about 500 samples, while other psychiatrists in the community supplied the rest of the samples. We published these results in various peer-reviewed papers. We also found that the NPR responds to successful medication treatments. The data for ADHD, shown on the left figure, were collected in our lab, whereas the data on the right were collected by the Johns Hopkins Psychiatry Department in an independent clinical trial. As can be seen, the NPR values are significantly higher in pre-treatment as compared to that in after treatment. L. Malik and others had already found that MP responds to lithium treatment in BD patients. The bar on the extreme right shows the data for untreated patients, while the left two bars show the data for treated patients and controls. All these observations are made using patients' blood cells, while mental illness is caused by the defects in neurons in the brain. How are the neurons related to the blood cells? Let us first understand the NPR in red blood cells. Gardos discovered the potassium channels in red blood cells as early as 1958. Hoffman and others at Yale showed that the Gardos channel is indeed the fourth member of the human soft conductance family of channels, or HSK4. It is also called KCNN4 in international nomenclature. These are calcium-activated potassium, or CAK, channels. They are widely distributed in the brain as well as in the peripheral cells. Are these CAKs really producing these NPRs in our experiments? I used the well-known potassium channel blockers, quinine and clotrimazole, to block the channels in the red blood cells in order to verify that the HSK4 do indeed produce the NPR values in our experiments. The blockers indeed block them as seen in this slide. Quinine data are shown in the left two boxes, while the clotrimazole data are seen in the right three boxes. This slide shows the schematic arrangement of the HSK4 pore structure. It involves 6 transmembrane segments as seen in the top left figure. The amino acid arrangement is shown in the top right figure. The bottom figure shows the side view and the top view of the potassium ion flowing through this channel. This positively charged ion flows through a negatively charged set of amino acids, producing electricity, just like a simple Faraday cell. Where do the differences in NPR observed in our experiments come from? First, I thought that they may be caused by mutations in the channel. I spent nearly 3 years to locate these mutations without much success. Then I turned to the signaling pathway controlling the CAK channels. There are 2 major pathways, namely CAMP pathway and DAG pathway. First, I tried CAMP pathway using CAMP promoters. There was no change in the NPR. Then I tried promoters and inhibitors of the proteins along this pathway shown in this slide. These proteins include protein kinase C, or PKC, calcium modulating enzyme, calmodulin, CAM, and DAG. Formyl 12 myristate 13 acetate, or PMA, is a well-known promoter of PKC. As you can see in this slide, the MPR values increase when PMA is added. Staurosporin is a well-known inhibitor of PKC. The MPR decreases in a concentration-dependent manner, as you see here. CAM kinase 2 is an important enzyme promoting CAM. Autocamtide 2 related inhibitory peptide, or AIP, is a promoter of CAM kinase 2. It also increases the values of MPR. DGKs attenuate DAG-induced protein kinase C activation. ALX is an inhibitor DAG kinase activity and increases the PKC activity. Thus, the MPR values are increased in a concentration-dependent manner as shown in this slide. The above results establish that the DAG signaling pathway controls the HSK4 channels in the MPR tests. The CAK channels are classified as big conductance, or BK, intermediate conductance, or IK, and small conductance, or SK, families. The SK family includes SK1, SK2, SK3, and SK4. The first three members of this family, SK1, SK2, and SK3, are widely distributed in the brain, whereas SK4 is distributed in the peripheral cells. The selectivity filter of the SK family is directly responsible for the selective and rapid conductance of the potassium ions. This filter is highly conserved in all the family members. This reflects the connection between the RBCs and neurons. Binding of a neurotransmitter to its receptor activates the G protein. Then the alpha subunit of the G protein activates the phospholipase C, or PLC, which cleaves the phosphatidylinositol 4, 5-biphosphate, or PIP2. The head is inositol triphosphate, or IP3, and the tail is the DAG. IP3 supplies the calcium to PKC, and the DAG activates the PKC. Thus, the neurotransmitter activates the DAG signaling pathway. The next slide shows the animation of this signaling process.
Binding of a hormone to a specific receptor in a cell's plasma membrane activates a trimeric G protein. The G-alpha subunit then activates a membrane-associated enzyme, phospholipase C. As its name suggests, PLC cleaves the phosphate-bearing head group of phosphatidylinositol 4,5-bisphosphate from its lipid tails. The head, now inositol triphosphate, or IP3, and the tails, diacylglycerol, or DAG, act as separate messengers. The hormone was the first messenger, and bind other proteins to affect cellular responses.
Let us consider the neuron as a radio transmitter for a moment. It receives the signal, processes them, and transmits them to the synapse. They sing in chorus. The disease symptoms appear when they go out of tune. Let us see how the DAG signaling pathway could be used for developing new drugs. Any one of these proteins along this pathway could be a drug target. Let us first consider DAG, which is a membrane-bound protein. A couple of decades ago, Hoken and others found that lithium increases IP3 and DAG in brain cortex slices in species ranging from mouse to monkey. This discovery produced a lot of excitement at that time and died down because people didn't know what to make of it. Stubbs and Agronoff found that cholinergic agonists such as carbachol would stimulate the formation of IP3 and DAG tenfold. Here is an example of reducing the required therapeutic level of lithium. Common therapeutic levels range from 1 to 1.5 millimole. 2 millimole is toxic. The left box in this slide shows the effect of 1 millimole lithium on MPR. The right box shows that the MPR improves with only 0.5 millimole lithium when combined with 2.5 micromole inositol and 10 micromole carbachol. Lithium side effects could be greatly reduced by the reduction in lithium dosage required. We do not know for sure if carbachol would cross the blood-brain barrier, although carbachol is approved for treating eye diseases. I looked for other cholinergic agonists used as psychiatric drugs. The well-known drug clozapine is a cholinergic agonist. It is recommended by UNESCO. I compared 1 millimole lithium result with 0.5 millimole lithium combined with 0.25 micromole inositol and 100 nanogram per milliliter clozapine. This combination shows significant improvement. The clozapine needed is below the therapeutic serum level of 300 to 450 nanogram per milliliter. Such a combination would greatly reduce the side effects. Aricept, or donepezil, is an acetylcholine esterase inhibitor used in Alzheimer's disorder to improve memory at serum levels ranging from 30 to 75 nanogram per milliliter. Compared to 1 millimole lithium, only 0.5 millimole lithium plus 0.25 micromole inositol with 10 nanogram per milliliter of donepezil would raise the MPR levels significantly as shown in this slide. So far we discussed how DAG could be used as the drug target. We could also use PKC and CAM as drug targets. While lithium increases the membrane potential, Ritalin, or methylphenidate, reduces the MP as shown in this slide. We know that Ritalin reduces the MPR levels in ADHD patients after successful treatment, as shown in this slide. We could use anticholinergic agents to improve the performance of Ritalin and reduce its side effects. Anticholinergic agents are known to reduce the cleavage of PIP2, reducing the formation of DAG. There are more than 100 potential anticholinergic candidates for this purpose. What we saw in this presentation demonstrates that the membrane potential could be used in the diagnosis, treatment, and development of new drugs by the use of the DAG signaling pathway. This pathway could also help in the understanding of clinical observations, just as blood pressure is used in heart disease. We are looking forward to transfer this technology to an organization with resources including people, lab facilities, regulatory know-how, and financial resources. It is our intention to dedicate it to the patients, their loved ones, and their caregivers.